Amino acids and proteins are the subject of this chapter, and let's look at the amino acid first. If you look at the amino acid structure, you can see there are two functional groups on it, and they reflect the name. There's an amino group and a carboxylic acid group, and notice they are both attached to the same carbon. The basic structure, and that's a bad word to use because when we say basic in chemistry we think of a base, but the simple structure of an amino acid is shown here. Any compound can be called an amino acid if it has an amino group on it and a carboxylic acid group on it, but the amino acids that we refer to in biology have this simple structure shown here where there's a carbon with a carboxylic acid group attached to it, and then an amino group attached to it, and then there is a carbon chain off to the side that we give a symbol R to. Proteins are polymers of these amino acids. Proteins are made of an amide link between two amino acids so that you have a carboxylic acid from one amino acid forming an amide with the amino group of another amino acid. And it makes a big long chain of amino acids all attached to each other. The amino acids that we're going to be talking about here are referred to as alpha amino acids because the amino group is on the carbon that is alpha to the carbonyl in the carboxylic acid. The carbon that is right next to that carbonyl is called the alpha carbon. The next one would then be called beta and then gamma. Now, we're not going to worry about that. We just know that the amino group has to be on the carbon adjacent to the carbonyl. There are 20 amino acids, and that side chain, the R group, differs for each of the amino acids. The amino acids do not exist with the carboxylic acid functional group protonated and the amino group unprotonated, because on the same molecule we have both an acid and a base. And you know when you mix an acid with a base, what happens? You get an acid-base neutralization reaction, where the proton from the acid comes off the acid and goes onto the base. And that happens inside this molecule. So if you look at this reaction shown here on this page, you can see there is a carboxylic acid group and an amino group on the left. The amino acid does not exist this way, but what happens is the proton comes off that carboxylic acid group and it goes onto the amino group, so that you have an anion where the carboxylic acid is and a cation where the amino group is. Because there is both an anion and a cation in the same molecule, this is called a Zwitterion. Some people may call it a Zwitterion. The nitrogen in the amino acid, in 19 of them, sticks out just like a primary amine. And you can see in the way that I have drawn the simple structure, the amino group is just stuck out there, not attached to anything. But in one amino acid, the R group is a carbon chain that comes back around and connects up with the nitrogen and makes a ring. That amino acid is called proline. It is the only amino acid that has something like that, so it's a little different from the others. The simplest amino acid that we have is glycine, and in glycine the R group is just a hydrogen. This means that that alpha carbon has two hydrogens on it. That's going to be important when we go back and talk about chirality. So glycine has a hydrogen as its R group. There are three possibilities for that R group. It can be a neutral chain, so it either has all carbons, or it might have carbons with an alcohol group, which is neutral, or it might have carbons with a thiol group, which is neutral. Or the R group can have an acidic group in it, a carboxylic acid. Or the R group can have a basic group in it, an amino group in there. The natural amino acids are chiral at the alpha carbon, except for glycine. Glycine has that R group is another hydrogen, and so you have two groups that are the same on that carbon. So all of the amino acids except for glycine are chiral, and all of the natural amino acids are what we call L amino acids. They're called L amino acids because when we draw the Fisher projection and we put the carboxylic acid up at the top, like we put the carbonyl up at the top in a monosaccharide, what happens is the amino group is on the left side. So those are called L amino acids. When we draw the Fisher projection, the hydrogen is always written on the right and the R group is always written down. The amino acid will exist as a Zwitterion only at specific pHs, and we call those specific pHs the isoelectric point. So for a particular amino acid, you'll have a pH at which it is a Zwitterion. If the pH is below this, then it's in acidic conditions, and acid will protonate the carboxylic acid, and it will be positively charged. If you are at a pH above this isoelectric point, then you're in basic conditions for that amino acid, and the proton is pulled off the amino group, and the amino group is uncharged then. Proteins are polypeptides. They're polymers of amino acids. When that polymer is formed, what you have is an amide group form. Let's look down here at how we can join two amino acids. One amino acid, functional group, carboxylic acid, will join with another amino acid, amino group. The enzyme that actually causes this to occur removes water from the two. So it removes an OH from the carboxylic acid functional group and a hydrogen from the amino group, and it joins it together and forms an amide. That amide functional group in a polypeptide is called the peptide bond. And this is showing how it was formed, and you can see there's a carbonyl from the carboxylic acid group joined now to the nitrogen in the amino group, and an oxygen and hydrogen from the carboxylic acid has been removed, and a hydrogen from the amino group has been removed, and we get this peptide bond. Peptide bonds do not spontaneously form. It is an uphill reaction. It can't even be formed without an enzyme, because even if you were to add heat in, it doesn't naturally form. When an enzyme does cause the peptide bond to be formed, it consumes energy, and the energy that it consumes is 4 high-energy phosphate bonds from an ATP going to an ADP. The peptide bond is planar, so the oxygen and carbon in the carbonyl and the nitrogen and hydrogen in the rest of the amide group are all in one plane, and they're rather rigid. So this gives a certain amount of rigidity to a polypeptide. The other backbone bonds are free to rotate but are chiral, and so we start having these polypeptides have varying specific arrangements. Let's go ahead and join two amino acids into a dipeptide. We're going to join alanine and serine. Alanine is the molecule on the left. It has an R group that is a methyl group, and serine is the molecule on the right. It has an R group that is a CH2OH group. Now, if we go to join these together, we're going to remove the water and form our peptide bond. you can see that the carboxylic acid from the alanine is going to join with the amino group on the serine. This means the amino group on the alanine is free and unbound, and the carboxylic acid group on the serine is free and unbound. They could actually form into other peptide bonds. The peptide bond is shown right there, and that peptide bond, remember, is very planar and rigid. The rest of the molecule can be floppy. Notice the alanine is written on the left and the serine is written on the right because the alanine has the amino group free and the carboxylic acid serine has its serine has its carboxylic acid group free. And that's how we always write our polypeptides, so that the amino acid on the left that begins the chain on the left has the amino group free, and then the last amino acid in the end of the chain on the right has the carboxylic acid group free. We write our molecules using abbreviations. There are 3-letter abbreviations. You can find them in your book in order to do some problems. There are 3-letter abbreviations that we use for the amino acids, and for alanine and serine, I've written them there, and I'm showing you how we can write a polypeptide starting by connecting the amino acids in order. Remember, the amino acid on the left has the amino group free, and the amino acid on the right has the carboxylic acid group free. I've written NH3 plus to emphasize that's a free amino group on that end, and CO2 negative to emphasize that is a carboxylic acid group free on the other end. We usually write them in their ionic forms like this rather than their unionized forms, because we know that's how it exists in nature.